in the Hackuarium lab with Marc and Adam. So if you want, you can have one of these cool.
Yeah,
This you can squeeze it out of that, and you can share it wherever you want. And yes, there's Vanessa Spirulina.
Oh,
Are you
nice.
doing well? I considered that.
I just thought I would just share.
There are a lot of spirulina in my life. I can't start to have loading. Anytime. And then when the show is over, you just throw it down the seat.
Oh, is that what you do? So when I
Look
was
out, you
surprised,
do. I don't know what to care about it.
we had this nice guy, David, talking about it the other day, and he seemed to say that when it's all fresh, it doesn't have that taste and smell of spirulina. But actually, I was scared to eat this. I didn't try it. But it definitely smelled, as it was drying, it smelled. Here, look. This is your spirulina, Vanessa. All dried down. But it's growing, the regrowing is going very well. So next time it bubbles, we'll point the computer camera towards that spirulina there.
A bit more light.
Can we just show them?
Oh, nice.
That little bottle right here.
Oh,
That's the spirulina right there. It's going to bubble eventually.
bubbling.
Because I don't know how often, it's like once in a while.
Great if the two of you have a bit of a dialogue.
Yeah.
Okay.
Oh,
Oh, yeah,
whatever.
let's have a dialogue.
You're
We'll
here,
talk
Marc,
about
so you got to do it.
genetics.
I'm doing some.
Okay, you're going to do some
So,
hacking.
No, I just
Let's-
it's not acting much.
you're
Let's
going
go.
to have a show too.
Yes,
Come on, show us.
I thought it.
Okay,
There,
let's
see,
take
because
the
we
big
have
one.
social
Take
distance.
the big
No,
one.
no, this is good. You can stay.
Okay,
Physical distance. Totally close.
so here we go. We're on number 3, and now we have 5 people. So hello.
Welcome
yeah.
to Hackuarium.
So we came to Hackuarium today just to talk about some labs that we're going to be doing this weekend on Sunday, and then 2 Sundays from this Sunday, and then 3 Sundays from this Sunday. So basically, 3 out of the 4 Sundays this month, we have 3 labs that are about how to make transgenic children. It's sort of a text-based, sketch-based, make art-based lab, but on the other hand, we're trying to actually show the genetic process of choosing a gene for non-scientists, for artists, for creatives, for people that are not trained in the language of science. We're going to introduce scientific papers, things like GeneBank or BLAST, like bioinformatics databases online, and make it fun to try to read or to read differently, to unread or to actually just sort of make the scientific papers your own. And our general idea is then to find a gene through imagining what that gene might be doing without even understanding everything that's said in papers, and starting to understand that the scientists studying these genes don't quite understand what's going on either. Like there's a lot of questions that it wouldn't be science if there weren't questions. So we're going to find our own ways to answer them, but we're going to also, by the time we get to our second lab, we're going to try to guide people to the sequence of the gene itself, which is not too hard, and download that sequence or copy it and paste it into a database. The database will start with the research in the first lab and lead to the sequence. And the sequence itself, you'll be able to insert it into a construct that if it was actually made, like synthesized, it could be mixed with a single sperm and injected into a single egg, and the ovum and spermatozoa would make a zygote that then would be transfected or infected or protein infected with RNA into the human genome. After that, I think our third lab is about doing bioethical reviews of what might go wrong, what might happen to the human that was born that way. And that will be how to become a bioethicist. And we'll also ask, how do you become an art critic? So if you're going to critique this new human that has a gene, possibly from a wolverine or a squid or a Tasmanian devil or like or pangolin, dandelion, a sweet pea, like spirulina. So what is it like to be a spirulina kid? And what are the bioethical things? But also, if we were going to say that these children were bioart, where it wasn't a bioethical problem, it wasn't a problem of science or enhancement, it was a problem of aesthetics, right? Like which gene from which spirulina do I choose for my child? It's probably an aesthetic decision more than a rational decision. And so people have to learn a little bit about the horrible business of art critique, which is like the biggest bullshit in the world. Take it from me, I'm an artist. Art critics are horrible. They're just ridiculous. So we're going to try that on, and also maybe there's some kind of open source contract for releasing your genetic research and your potential human genome infectious agent and your bioethical review and your arts review to release it to the public database, which will be open source. Uh, you have to sign a contract, right? Like a Creative Commons contract. But we also want to teach people how to not just hit I accept. Because when you say I accept a contract without even reading it, without especially editing it, you're actually just signing a blank check, right? Mostly for your privacy. In this case, we just want to teach people how to unwork a contract a little bit. And so we have a lot of art projects planned in between the lines of these goals. And it's actually supposed to be fun. I have a funny feeling it will. We're breaking in with some performances, and we have a lot of loud people that we're inviting. And so I'm kind of looking forward to sharing it. And I wanted to invite the Hackuarium crowd and anybody out there. Are you out there?
Yeah,
Um,
we hope so.
yeah, to join either online or in person. It'll be in Zurich, but also we're streaming. And just, feel it out. I think the labs will be quite intriguing. Um, we're still working on something called the GOSHPA, which is the generic open source um, plasmid
Plasmid.
plasmid for human
You too.
alternatives. Or I like the acronyms. I do. I can't even explain them myself. But basically, we're trying to make an open source plasmid that you could drop any gene in that, if it was synthesized, it would be useful for making a transgenic baby. We're not synthesizing it today. We might take the best of and get them synthesized, and we wouldn't start by making babies. I don't believe so. There's a little,
Make an internet.
there's a little more than licensing involved. I have to say, as a parent, but we might, look into whether or not we could utilize this generic open source plasmid for, I don't know, maybe we could just check out if it works. Like get some human embryonic stem cells or some pluripotent cells or something like that, and sort of you know see if it actually has any effect, right, on these cell lines. I think the licensing for that, if to do it legally, I think you would only have 7 days and have to come up with like some reason why it was science. We want to do it for art, so that makes it even harder. We might have to consult with some sort of bioethical art advisory commission or something like that. But right now, the lab is text-based. It's kind of friendly, and it should not just demystify the process, which is important, but also not totally promote the process. And in particular, it's meant to not promote the process in the name of making better children. So these are not designer babies in the line of transhuman enhancement and perfectionism, but actually looking at the biodiversity in the world and the number of genes available throughout the entire ecosphere, and then looking into other aesthetics than making us purely better, because I don't think purely better is a simple concept, right? It sparks of, like, it sounds like neo eugenics. And so we're basically sort of like offering, if it's going to happen, we need to have sort of a queer database of other options, right? Than sort of like monocultures of sort of supposedly perfectionist children that turn out to be old yesterday's model within a couple of months, right? How does that sound? Is that a good blurb?
It sounds bad. It
Yeah.
sounds really bad. This is the kind of biohacking that we are not going to do in this lab. But the point is to give a forum to discuss all these problems because when you look at the Chinese designer babies, how are those little girls doing now? I
Lulu
wonder.
and Nana, they're hiding them behind patient privacy, but they're not just patients.
They're people.
They're people, and they weren't born for a medical industry study, but I'm sure that their parents signed them up for 18 years of poking and prodding. And the fact that they're anonymous, uh, I think it's misusing patient privacy
Mm.
because I think that, in particular, maybe Child Protective Services has to get involved.
I wonder if that's possible
You know
where
what I
they
mean?
are, of course. It's
It's
not
a.
the same as being back in America.
I'm not suggesting anything over in Shenzhen, but like I'm sure they're out there, And anyway, I think of Lulu and Nana a lot. In fact, this lab is sort of dedicated to them.
They must be at least 3 years old now, right?
Yes, and their technical, like their techno father,
Mhm.
the scientist that helped get them injected with his desires,
Mhm.
he gets out of jail around the time they turn 3.
Oh, really?
Yeah, he'll
So
be
we
out.
should keep track of that.
I think they're going to all have a birthday together, so maybe we should go celebrate. Maybe give him a sculpture trophy. Very good sculpture. Both of them, No,
It
he's.
would be really nice to sort out the whole story because there was this thing about were they actually just made mutant so they couldn't be infected by the AIDS virus, or were they made mutant because somehow that might be related to better cognitive abilities in mice?
So by mistake, in the name of health, right, which is often the
Mhm.
excuse in science, like you get more money to study Alzheimer's than to actually just do pure research.
Mm.
So people say, I study Alzheimer's, but in this case, this gene happens to give some immunity to AIDS, possibly, and it's known to have some sort of strong analytical ability, right? Now, I'm I'm like Russian Jewish, Ashkenazi Jewish, and we have a gene in us that protects us sometimes from AIDS. I don't know if it's the same one, but we also have unusual analytical ability. But I would say that with that comes some detriments. We know that the AIDS resistant gene makes you more susceptible to the common flu.
Uh-huh.
But analytical ability, in my opinion, makes you more susceptible to total disassociative, what would you say, neurosis, right?
Mhm.
So maybe we didn't enhance them. We made them more susceptible to the flu and more neurotic, which I'm sorry.
Hmm.
That wouldn't go, that doesn't get reported quite so often. So
Mm.
this is what we're talking about bioethics, like this sort of idea of let's look into it as creatives, even if we sound ridiculous, like a little conspiracy theory. The truth is that not only is the truth not really out there in the news, etc., that there's things to uncover that we don't know about these kids, right? Like were they funded by? Actually, we know this. They were funded
Mm.
by international, you know, people that happen to surround the Human Genome Editing Initiative, right? Big players from Stanford, from Rice University, from MIT. I think we know their names. And like, what other kind of funders were funding the incubators and the startups that led to this? And how did the Human Genome Editing Initiative kind of set the stage for this to happen, right? And was it I don't know, subtle racism to have it be done first in China, right? So there's a lot of questions. Um, I don't know. I think Lulu and Nina need to be addressed. Maybe we'll do a little address in these labs, like sort of like, hi, we know you were built. We were built with help from MIT and Rice University and Stanford University and the Human Genome Editing Initiative. This rogue scientist in China made you, but you're part of a bigger program. You know, Many people have been born this way that haven't been announced on the news,
Mm.
right? These babies were planned to be born on the like 5 days before the Human Genome Editing Initiative met in Hong Kong. That takes more than 9 months of planning. So it was a PR stunt. How does it feel to be born as a PR stunt? Right? That's confusing. So this is the kind of questions you're asking too, yeah?
Yeah.
Yeah.
But I mean, I've never done anything with CRISPR myself, but I've always thought it would be super to use as with the non-cutting version for labeling bits of the genomes. Where I'd really like to look at for this dynamic genomic integrity idea,
Mhm.
look at how subnuclear architecture is moving around. And if you had like
Right.
fluorescence tag on chromosome 2
Yeah.
and another color on chromosome 5, and you could watch it live, it would be
Sort
really
of like
cool.
real-time tracking.
Yeah,
Yeah.
yeah, yeah. And
That's
after like exposure to various things we know that are genotoxic.
Now, how do you, this is a confusing thing for genotoxicity.
Mhm.
How
So
do
you
you,
want to make a kind of marker baby.
No, I don't want to
So
make a
it's already kind of, it will just glow weird when this genomic integrity goes forward that the little pinky starts to glow or something.
No. I'm more into totally unmodified people and using the inner cheek cells
so you don't
to
want to
see
use,
if the DNA is
you don't
strong
want to
enough.
use people as environmental toxicology markers.
Only parts of people.
Because I mean, there already are, there is that. You could have, like, for instance, if you look at cancer maps, right? Cancer maps are showing where people are dying of cancer from environmentally toxic environments, right? Or
And you
places.
would think they'd do something to clean up those places by now, but instead they just, I don't know, they
They just
they
map.
make lower pricey housing, and for these poor people that live there, they're getting really.
It might be nice to make an eco marker baby. Like, if there's too much BPE in their system, maybe they do start to glow a little. You know, that's interesting.
We're gonna make a baby that's just one giant cheek cell.
What giant cheek cell? Oh,
Then we don't need the baby.
Whatever.
so this gets interesting. I don't know. We're planning on having some performances too, right? And there's more art to this than you'd think. Like, I might demo how to cut up an abstract, like cut it into quarters and rearrange the abstract and then reread it. You know, to see what it says when it's been taken out of order. So this is kind of, there's some making fun of science and making fun of reasoning, because it seems like this project might not just be about reason.
Well, critical thinking should always be fostered. That's what I say.
And so we want you to get involved also on many levels. Huh? So when we'll go through this process, we also put stuff on the wiki. We have the forum. We have the workshop, which is just like this kind of focused time slot. That of course you can collaborate throughout the next 9 months minimum.
Definitely.
And we could do another session here later in the project to kind of maybe even present a bit the progress we have made through the last, or you could also join and kind of give your own critical review of our genetic construct we produce with the participants. So this is more the role of collaborators that deeply get involved and kind of give feedback, and then
That's one of my areas of expertise, just that sequence gazing. How does this fall? Where's the pseudo knot? Yeah,
Really? So this is something you do? Oh,
we have a new role, sequence
sequence
gator.
gazer. That does sound good.
But some people don't like that term.
Yeah, I like it a lot. One of the things that we're working towards is a sort of a creative construct genome database, right? Like if people do some research on a gene, even if it's normative, and then they actually find that gene sequence and they enter it into this sort of open source plasmid and it's online, and they add a bioethical review and a bioart review, then as long as they sign some version of some contract, or maybe if they don't even, it'll go into an online repository, like a database, a bioinformatics database that is, I guess you would say this is a most alternative database for the future of humanity, right? So we're looking for genes that are really obscure. We're looking for people that are engaged enough to sort of wiki hack the human genome, right? And we want to make like a sort of pertinent impact by having all of these public and making it so that if people wanted them in the future, they can see when they got made, how they got made. Well, it wasn't quite determined by corporate funding or state industry, et cetera, or the military, but was actually like self-organized and
Just
self,
personal interest.
yeah, sort of maybe even, I guess you would call it transgenic human brute or transgenic human naive art, you know? But in some ways, we may be offering one of the only options, you know, to what would otherwise be a sort of military industrial enhancement neo-eugenic project that is going on. It's not over, it's just beginning, you know. This kind of thing.
Let's hope not.
Yeah.
In some oligarch's yacht somewhere, they're probably working just on their, cosmetic surgery things. You really think they're doing things like that?
I think in the IVF clinic down the street, it's already offered. I think there are IVF clinics that are listing.
I think they're planning ahead towards, there's definitely a lot of like long-term strategies and planning, and they come up with concepts when it's technically and legally at the moment not so possible. But if you run an IVF clinic for 150 years or like 70, 50 years, you have to make sure you're ready for. So they have this kind of plan for this long term, and they already have this think tank, and they have a list. They already have.
If you look at the interface between new reproductive technology and sort of like culture, right? So like if you're people in India will pay for an ultrasound just to sex their embryo to see whether to get an abortion because they want a male child. That's a complete misuse of ultrasound. But like IVF was considered to be, oh, you know, sort of scary when we had our first test tube baby. And now it's like 1 out of 6 people in Australia is using IVF.
Really?
And sometimes it's because they want to, you know, they want to time the birth of their child to when their parents aren't available. You know, like people do things for the wrong reasons, you know. But I would say, you know, I mean, there's various different sci-fi scenarios, but it's not sci-fi anymore. It's like we know that it's possible. We know that people have made them, and the fact that it went public means that it's happening more often. There are a lot of labs that are doing work with human embryos that are viable, but not human embryos with trisomy, but human embryos that are viable, and they have, you know, 7 days to 14 days to officially to not implant, to actually terminate those experiments, right? But if they're born again Christian, they're going to implant them out of an honor to, you know, what they believe in. They need to implant those snowflake transgenic babies, you know, because otherwise they're causing harm. After they're born, they can go to jail forever, but they must be born. Anyway, so yeah. What do you think? You think we're making it up though?
No, no, I think the whole in vitro fertilization is out of control. It's very clear that oversight is rarely possible when you're in these private clinics.
Mhm.
People are making choices left and right, and if really 1 in 6 people,
Well, you have to understand that the number of people that came to Australia to begin with was an island nation. I'm talking about the imports, you know, like from the UK and surrounding areas, those prisoners. There was
you're saying
really they only had like, you know, 16. No,
not enough of a gene.
yeah. So I mean, well, there is a lot, you know. I mean, there's a lot of because of that, maybe cystic fibrosis, Down syndrome, etc. And after, you know,
I mean, it would be so amazing to cure genetic diseases, right? But all the things with viral vectors, you always ended up with some mess because there was an integration site you didn't know about, and then they have leukemia.
yeah.
Or so you give up on that vector, you try another one. Like AAV now is even considered, you don't even need P2 for it anymore.
Yeah, but this is confusing because AAV was, I think, how Jesse Schlesinger, what is his name? Like, I mean, wasn't it how the first case of gene therapy causing leukemia came about?
No, no, I thought it was the lentivector.
Oh,
It
that
was
was
a
Lindsay.
lentivector.
Anyway, I have looked and there are some random, semi-random, you know, genomic integrations of AAV, but this is one of the things we're talking about in this lab. And one of the things that we're talking about is, first of all, we know that CRISPR is more targeted than random integration into the genome with some kind of virus. But that doesn't mean that it's targeted well. And there's advances into it, but where it doesn't target well, it can cause, sure, you know, it can cause cancer. But besides that, it can break whole chromosomes in half. You know, like there are off-target mutations, and then there's chromosomal breakage. It's not as accurate and clean as it's claimed. That's sort of ridiculous. But it's more accurate and more targeted than no accurate targeting at all. But besides that, there are places in the human genome that have been determined, or at least they're labeled as safe harbors for CRISPR landing spots.
Mm.
And this concept of a safe harbor in the human genome where something could land and it doesn't matter, it doesn't do any harm, is still really up for question. I mean, it's
No, they have no idea.
it remains to be seen whether it's a safe harbor, right?
Right.
Yeah. So yeah, that I believe
And it's clear, I mean, our chromosomes are doing a lot of stuff. Like we wouldn't have a real immune system if we didn't have recombination and breakage and repair with mistakes.
Mhm.
So we can recognize all the things we encounter in our lives.
Yeah, it's almost like this random generation thing, right?
It
Like a
is, it
generative
is. I mean,
algorithm.
that's what life is about, is maximizing differences sometimes, right? And
Yeah.
when a person decides they know what's the right thing, usually they're proven wrong.
So this is one of the things I'm looking into is what are the, top 20 safe harbors so far? Because if you pick the top 20 safe harbors in the human genome to land a new construct, a new gene from another organism, etc., there's a question there. Out of those 20, which one is not a safe harbor at all? Which one's going to cause some kind of downstream detrimental effect? But besides that, we also have to look at promoters, right? Like, for instance, you might not just want this gene to be on all the time, even if you picked something from a moss plant and you think it might really help people have more chest hair or nicer skin or mossy ears or easier to shave because their hair is mossier. You don't want that on all the time or you'll have super moss, right? And in fact, you can have it express, a certain amount. You can have overexpression, underexpression, demethylization, whatever. But besides that, this is sounding really strange, but you can have external environmental effects that promote the gene construct in the human body. So that means that someone could have a gene from another organism put into their genome and be born that way, but it wouldn't get expressed unless they rubbed butter on their forehead. And all of a sudden, those forehead cells would pick up the butter and say, "Butter forehead, turn on the foreign gene. Turn on that transfection. That transgene transfection." And so we're looking into sort of different safe harbors and different, I don't know, environmental factors that might cause the gene to get promoted, overexpressed. So, I don't know. Can you think of some interesting promoters that we might, you
Well,
know?
I think it's always best to target your mutational change you want for your special designer baby right to where it belongs, actually. I would say you want to use the endogenous promoter.
Uh-huh.
So you would make something where, like, there's a mutation and you want to fix it.
Uh-huh.
And then that would be driven by its ordinary promoter. There's not going to be any overexpression. There's not going to be any real transgenesis even. There's just going to be fixing. But it's dangerous.
So,
I know, that's
so
why.
bring the human species to its older form, like Homo erectus.
To fix us.
All the
Yeah.
stuff that happened on
Well,
the
his frontal lobes
That
are
means
really
it
rough.
makes the breast cancer gene not work to repair DNA. You would fix it so again it works.
But your
But there's
baby
a lot of
doesn't.
possible
But your baby
mutations.
doesn't yet have breast cancer. So
No,
It
exactly.
has a statistical propensity for a BRCA2 gene that might cause breast cancer later, and you're actually, I mean, would you turn off their breasts
Well, I
before they were
mean,
born, to save
I
them
would
from?
say then you want to keep them away from anything that could damage their DNA. But the problem is that things happen inside of ourselves that are
Yeah.
damaging DNA all the time too, and we can't
And then there's
really stop everything.
butter on your forehead causes cancer. I think we know that. But I mean, yes, it is a problem of purity and a problem, but oftentimes I believe in gene therapy to fix detrimental disease, but I also believe that a lot of gene therapy experiments prey a little with informed consent on people who have lethal, and emergent desperate.
Call for desperate measures.
And then, but the larger project is not to fix genes.
Hmm.
It's to make people live longer, grow taller, have bigger brains,
Pay money.
yeah, pay a lot of money, have bigger breasts, a more aquiline nose, very fair skin, and probably giant penises. Um, this is a problem to me because that's like 7 kinds of enhancement and it gets boring. I understand that you can knock in a fixed gene over a broken gene, but we're talking, well, that's knocking out, right? You knock out the broken gene and you knock in the fixed gene, but you could possibly knock in genes from anywhere and you can put them endogenously over genes that are detrimental to humans. Like, for instance, there's a gene for gullibility. Maybe we'll just cover that up.
That's a multi-gene
You think that's a multi-gene tree? Damn, we can't knock that out because that would really help. That would really help a lot of people
What is gullibility?
when you believe anything.
You believe all the conspiracy theories and the fake
Oh
news.
man. So
So maybe we should see if anyone in the audience has any questions. Is there anybody actually watching this?
So many.
Should I look on the juggle?
well, I see that we got 3 thumbs up, but that was from one person, and that means it's a person with 3 thumbs.
Maybe they've already been playing with your
Yes, there are
stuff.
3, yeah.
Is there anything there in Jitsi? No, Jitsi's still just like that. So there we are. We're live. Well, it's at least it's more stable than when we
It
were
is.
outside.
This was try number 3.
I think we can also maybe go off live, you know, and hang out, give it
Yeah, well, we can
or
do
to
a
a
little
photo.
tour.
Yeah.
We can just show people. Here's the lab.
Yeah.
This is everything. It's a beautiful picture.
Oh, and by the way, you can show up online on Sunday. You know, come to Hackuarium. Come.
Come to Zurich and Hackaria.
Put the link in the
Oh yeah, we can put the link in the thing.
I'm glad to talk to you folks. This is, I'm in the Hackuarium. I'm going to do Hackuarium swimming.
You want to meet the mascot?
Yeah.
We've got several. Here's one.
Oh, come take an octopus gene and put it into your transgenic baby.
And make it resistant
Oh, this is excellent. Oh, this is the octopus. This is the mouth. This is nice. Ooh.
Here's another one.
Um,
Whenever we have little kids
the
coming
name Hackuarium and the octopus logo is kind of hard to beat, man.
Somebody told me it's the perfect logo, actually,
Ah, it is.
too.
wow.
And these are very nice, huh?
Oh.
So shall we juggle the coronavirus?
Not bad.
I could talk about the urban gardening project. As you can see, we are cultivating our inner Gregor Mendel. We have 2 plants here of 2 different groups, the A group and the B group. One of them has the bacteria that make a symbiosis in the roots of the plants, and the other one was just mock inoculated, doesn't have the bacteria. And so there are 16 sets of people with these plants in their gardens, in pots. But this is the first year for this urban garden project where we decided we would just have some growing indoors the whole time. And check it out, the first peas are already coming.
Which one is with rhizobium and which one is
I
without?
can't tell you, it's A or B.
It's
I'm
one
not
of
going to tell you.
Oh,
I can't tell you until the end of the year.
I can tell. I can
Yeah,
tell.
look at that. There's
Yeah.
already 3 nice peas here, and there's flowers coming up there too. Look at how tall it got too.
Oh.
And I have to switch them around to change the sunshine.
You know, what's it called when you're doing an experiment and you already can tell that you have good results, and out of pity to the other people with the placebo, you go ahead and like give them the medicine?
Oh, that's exactly what happened
feel
with
like-
Aerosmith.
Yeah. That's interesting. Calm down. Yeah.
It's such an ethical quandary.
So I think I'm going to pirate some rhizobium into your one without pea pods.
Oh,
I
that's
feel so bad. This is so mean. I'm sorry. I'm sorry, A. I gave it away. I gave it away.
So those are the ones. So look, there's also a special experiments about the different kinds of soil.
Oh, nice.
So last year, our surprise result was that the plants that were in sort of hydroponic conditions, they turned out to be worse, way with the bacteria than without.
Okay.
And
Because hydroponics like
I
also a little stressful,
I
yeah?
don't know. But so this year we have more plants in hydroponic conditions too. So this is the set that's not getting fed. At home, I have some that are getting their special
This is so
food that we
cool.
normally get. And we can go for a walk over by the school. We have a couple of plots of land that have pea plants in them too.
All right. I'm going to pass this back.
Okay.
Well done.
So now you want to have a little, you want to do some bio art, some sort of. Have we done enough?
I think we should get our private tour to Hackuarium.
Okay, we have to do the private tour now, but leave a message and we will come back and we can even try for try number 4. Thank you.
What is this?
This is your cleaning stuff.
Where did you get that?
Gustavo had it. Here, I'm going to give the peas some water. I said I was going to do this before. And I want to